Hi, how are you? I hope you are doing good. So, let us begin with our next unit. And this unit deals with the role of calcium in cell death, role of calcium in cell injury, role of calcium in different cell functions. Calcium, you know very well. Calcium is a essential element like sodium, potassium. Calcium is taken by the body through diet, by calcium-rich diet. We take milk, we take egg, we take vegetables, we take cereals. They are all rich in calcium. So the source of calcium is diet. And extra calcium, the calcium goes to intestine, it is absorbed, and the extra calcium is excreted through feces. And the remaining absorbed calcium is taken to the blood, and from blood it reaches to bone. So, bone is the major depot of calcium in the body. Uh, if you just calculate in percentage, 99% of our calcium is in the bones. Therefore, the bones are so strong, and 1% of the calcium remains in the body fluid. Uh, Calcium concentration inside the cell is higher, and calcium concentration outside the cell is very low. Say 100 nanomoles is present inside the cell, and 1 nanomole or 2 nanomoles are present outside the cell. In addition to bones, calcium is a very important component of the cytoskeleton. And if you just ask me where the calcium remains in the cell, it is bound by the proteins of the endoplasmic reticulum. It remains bound with the endoplasmic reticulum. It remains bound with mitochondria. It remains bound with the plasma membrane. So calcium uh, is a very important, or the subject of role of calcium or toxicological consequences of calcium is very important. We talked two very important events. We talked about the free radicals, lipid peroxidation. We talked about oxidative stress. And now we are coming to a very important issue in toxicology, and that issue deals with calcium. So the calcium ions, they play a very important role in different mechanisms in the cell. They play a role in signal transduction mechanism. They play a role in membrane integrity or membrane fluidity, and they play a very important role in gene expression studies. And finally, they play a very important role in cell death, whether it is apoptosis or whether it is necrosis. So, uh, who controls the calcium in the concentration in the body? The calcium concentration in the body is controlled by hormone, by a hormone, and that hormone is secreted by parathyroid gland. This is the place of thyroid, and adjacent to thyroid there are parathyroid glands, and these parathyroid glands, they secrete a hormone, and that hormone is known as parathormone. If you just cut the sections of parathyroid gland, you will find the it has got the gland cells, and those gland cells, they secrete an hormone, a hormone, and that hormone is called as parathyroid hormone. So through a feedback mechanism, the concentration of calcium in the cell is maintained. Whenever the cell needs calcium, the signal goes to bone, and bone releases calcium into the circulation, and the calcium reaches the cell. Whenever when the pool of calcium is full, there is no signal coming from parathyroid gland in the form of parathormone, and the calcium entry into the cell is blocked. So this is how, with the feedback mechanism, with the help of an endocrine mechanism, the calcium concentration in the cell is maintained, which is very important. If the calcium concentration increases, I was telling you about blabbing, membrane blabbing. the blabbing starts, and the blabbing starts with the disruption of the cytoskeletal components. So the calcium, excess of the calcium leads to membrane blabbing, and excess or the increased lipid peroxidation will result to reduced to an increased oxidative stress, and oxidative stress will result into a higher amount of calcium in the cell. Though calcium plays no important role in oxidative stress, it is not a redox active element. Remember it, it is very important. Which is the redox active element? It is calcium. It has no role to play in oxidative stress, but it has a very important role to play in cell injury. The people have worked for last 30-40 years on role of calcium in cell injury alone. I can talk to you about Sten Uranius, who comes from Karolinska Institute, Sweden. He has devoted his entire life to study the role of calcium in cell death. Right? Similarly, there are other people who are working, and they have mainly, Oranians have mainly worked on acetaminophen toxicity, AP toxicity, and they have delineated the pathway by which AP through a reactive metabolite, through a toxicophore NAPQ1 uh, is responsible for causing the toxicity. So this is how you see the sodium, the potassium, and calcium, they are essential elements. But the calcium has been found to be very important in cell death. When we will be discussing about necrosis and apoptosis, we will discuss one type of necrosis which we call as coagulative necrosis. And the reason for coagulative necrosis is the precipitation of the cytoplasm by excess of the calcium. It's just like the formation of a curd. As you see, the fermentation occurs and the curd is formed, in the same way the coagulation occurs in the cytoplasm and the necrosis follows. It is called as coagulative necrosis. We call it as coagulative necrosis. So that is calcium dependent. We call iron dependent necrosis as ferroptosis. That is called as a new term is coming up, that is called as ferroptosis. There is another term coming up that is known as pyroptosis. So now it is not only apoptosis or necrosis, there are other types of cell death which we can discuss at a later stage when you are mature enough to understand those events. Then we can discuss the ferroptosis and the pyroptosis. So at present we should discuss calcium, and I told you it is not a redox active element. It has got no role to play in oxidative stress, but it has got a, so calcium mediated toxicity is not oxidative stress mediated toxicity. You should be clear about it. Then what are the regulatory mechanisms of calcium? Calcium, if you remember, the anatomy of thyroid or certain physiological aspects of thyroid, you find that thyroid also secretes a hormone which is known as calcitonin. Calcitonin, like parathyroid hormone, calcitonin also plays a important role in regulation of intracellular calcium levels. Similarly, there are certain proteins which are accountable, responsible, which are attributed to calcium, and the calcium is sequestered by those. Calcium is, you see, scavenged by those proteins. They are known as calpains. So whenever the higher level of calcium enters into the cell, the calpains try to squeeze it, try to sequester it, and that is with the help of another molecule that we call as calplastins. So these calpains are calcium dependent proteins. Calplastins are the molecules which work as a feedback mechanism. So you must understand these events, that the calcium which is released from bone comes to blood, goes to the cell, as and when required, is also when excess of the calcium is present, it is sequestered by certain calcium dependent proteins which are known as calpains and calplastins. So in addition to that, I would like to discuss with you the few things, that there is a membranous at the plasma membrane level, there exists a pump, and it is just like a sodium pump or potassium pump, and we call it as the calcium pump. And this calcium pump works in the mitochondria, and it plays a central role in the cell, and it also plays a important role in the efflux and influx. Efflux means going out, influx means coming in. Efflux and influx of calcium. So there was an experiment which was done by certain people, and that experiment was done with tributylene. Tributylene is a molluscicide, and that mollusk dentalium, you must have heard about dentalium. Dentalium is a marine mollusk, and it lives inside a shell, but it is a parasite on wood of the ships or the boats. So the boats which are anchored into the marine water, into the brackish water, they get infested by a molluscicide, and that is the tributylene. And tributylene has a very important function, that it inhibits the calcium influx into the cell. It inhibits the calcium influx. No, sorry, it increases the calcium influx into the cell. But there is another molecule which is EGTA. It is a chelating agent. If we treat the rat or experimental animal with EGTA, the calcium level goes down, like EDTA, like penicillamine, like British anti-leukocyte, like other chelating agents, Ball and others. This is this works against the accumulation of calcium into the cell. This is EGTA. Then there is another very important it works against the penicillin. There is another very important issue, and that deals with another calcium dependent enzyme. There are some calcium dependent enzymes. One is the caspase. Caspase plays a very important role in apoptosis. On and off, we measure caspase activity in our lab. So whenever we are trying to find out to confirm whether it is causing apoptosis or not, we take the help of this enzyme activity, caspase. This is the cysteine dependent enzyme, calcium dependent protease. This is present in the cytosol, and this calcium caspase activation leads to apoptosis. These calpains, which are calcium dependent cysteine proteins, they are responsible for cytoskeleton. Say for example, spectrine, vimetine, keratin, and casplastine is the inhibitor of the calpains. I told you it works as a feedback mechanism. Calcium has got a very important role to play in signal pathways, signal transduction pathways. Calcium plays very important role in the damage caused by the acetaminophen, and calcium can activate the endonucleases. Calcium is responsible for phospholipases. I might have discussed with you these phospholipases. And there again, it is a family of enzymes: phospholipase A2, phospholipase B, C, like this. And these phospholipases are responsible for the deterioration or denaturation of the phospholipids. and the polyunsaturated fatty acids. It is very closely related to lipid peroxidation, and it leads to death. Another very important issue related with calcium is the mitochondrial injury. You see, nowadays I'm reading very extensively on mitochondrial toxicity, mitophagy, mitochondrial homeostasis. This is an opportunity for me to read all these, to study all these topics which have not been studied earlier. So I found during my studies that the mitochondrial calcium is a very important function or very important subject. This is there is a calcium and sodium dependent enzymes, ATPases, and the ATP depletion occurs during many toxicological conditions. So there also the calcium plays a very important role, and then calcium plays a very important role in gene expression. June, FOS, MIS, these are all related to the death receptor pathways, and the activation of these genes leads to apoptosis. So this is how calcium is playing a very important role in cell injury and cell death. The toxicological consequences of the increase of intracellular concentration of calcium are are many, and these toxicological consequences are very important, and they need to be studied at length. That what are the toxic, say we I have taken a lot of calcium, doctor suggests that you take vitamin D and calcium, your bone density is very low. Generally in the people of my age, those who do not take sufficient amount of yogurt and milk, they suffer from calcium deficiency, and the doctor suggests that you should take calcium rich diet. And that calcium rich diet can, or the excess of the calcium, you see, it is a double-edged sword. If you take a higher amount of calcium, it will be toxic. If you do not take calcium, you will suffer from calcium deficiency. So the disruption of intracellular calcium content leads to very important events in cellular biochemistry. It can lead to irreversible damage. Increase in cellular calcium level can lead to irreversible cellular damage, and the consequences of the intracellular increase in calcium level are like activation of calpains, like effect on nuclear endonucleases, like activation of calcium dependent phospholipases, calcium cycling in mitochondria, and then the gene expression studies. This we can take maybe in the next lecture. I will deal with the consequences of higher concentration of intracellular calcium. So study well. This will form a background or a platform for you to study the toxicological consequences of higher intracellular calcium concentration. Bye.